The discovery of the complex processes of translation is an accumulation of the work of several scientists across the different disciplines of science over the past century. The first major breakthrough in finding DNA structure came in 1950 from an Austrian scientist called Erwin Chargaff. Chargaff discovered that there was a 1-to-1 ratio between the amount of guanine to cytosine and the amount of adenine to thymine in a cell, i.e., the percentage of adenine equaled the percentage of thymine, and the percentage of cytosine equaled the percentage of guanine. This became known as Chargaff's rule. American chemist Linus Pauling played a role in the discovery of DNA structure when he determined the helical structure of proteins in 1951. Although his suggestion of the triple helical structure of DNA was wrong, it paved the way for English scientists Watson and Crick. James Watson and Francis Crick built on Chargaff's and Pauling's discovery, and along with it and the X-ray crystallography by English scientists Rosalind Franklin and Maurice Wilkins, they developed a model of the structure of DNA. Their model was based on the diffraction patterns of DNA captured by Franklin, which suggested that DNA had a helical structure. They showed that DNA was a double helical structure with a right-handed turn. They also proposed that adenine forms 2 hydrogen bonds with thymine, and that cytosine forms 3 hydrogen bonds with guanine. This coincides with the helical structure and abides by Chargaff's rules. This was a huge discovery and really opened the doors in the study of molecular biology and genetics. Crick continued to work on DNA and developed theories on protein synthesis, which he proposed in 1958. He coined the term the central dogma with his theories. The central dogma is the flow of genetic information from DNA to RNA to protein. In 1961, Marshall Warren Nirenberg cracked the genetic code. His initial experiment was known as the Poly-U experiment. A synthetic RNA molecule made purely of uracil was placed in an environment with ruptured cells. The cell's contents were free-flowing with the protein synthesizing components free to interact with the RNA. He found that phenylalanine was produced. This was the first step in cracking the genetic code. He continued to work and decipher what codons produced what amino acids. We will now discuss the molecules involved in translation.
Molecules that I would like to talk about are ribosomes and tRNA. Ribosomes consist of a large and small subunit. In both prokaryotes and eukaryotes, ribosomes have the same function, but differ slightly in size. Small subunit in prokaryotes is 30S and large 50S. In eukaryotes, 40 and 60S respectively. Each subunit consists of proteins and few ribosomal RNA that are most of the mass of ribosome. They are synthesized in the nucleus and released to the cytoplasm wherewith mRNA form functional ribosome that is capable of translation of proteins. Each ribosome has 3 sites. A. The point of entry for tRNA. P. Binding site for peptide, tRNA and growing peptide chain. E. Exit point for uncharged tRNA. tRNA, transfer RNA, is a short, around 80 nucleotides, single strand, folded on itself. Formation of hydrogen bonds between complementary bases form its L-shaped three-dimensional conformation with 3 loops, one which is anticodon, sequence of 3 nucleotides that corresponds to the codon of mRNA coding for specific amino acid. 3' end of the strand is a place of amino acid attachment. Amino acids form covalent bond with 3' end of tRNA. This process is catalyzed by amino acid tRNA synthesis specific for each amino acid. This process is conducted repeatedly and tRNA molecule is used more than one time. tRNA with attached amino acid is charged. It functions as translator of mRNA. It has the ability to read mRNA and match anticodon at the end of the loop with mRNA codon.